This is a video of the genetics summary, and I'm going to do it in two parts. So the first thing we started with when we looked at genetics after doing the activities was Mendel's study of pea plants, what he did and what he found. We wanted you to know a bit about the structure of a flowering plant, that the male part is the stamen, the female part is the pistil. The male part produces the pollen that contains the sperm, and the female part has the egg. And so pollination is what happens when the pollen reaches the pistil, and fertilization happens when the sperm reaches the egg. Now Mendel's pea plant started off creating the P1 generation by doing self-pollination. So that meant that the pollen from the same plant fertilized the egg, and over generations you end up with a purebred pea plant, which we would now call homozygous. And then in his later studies he wanted to ensure cross-pollination, so he removed the male part of the stamen, and that ensured that the pollen arriving was coming from a different pea plant. So he could do a lot of controlling of that. The observations were from the P1 to the F1 to the F2. P1 was the first generation of the purebreds. The F1 was the result of that cross, and they happened to all have identical heterozygous genotypes. And then he thought, what will happen if he crosses that? And that gave us the F2 generation. Principle of unit characters. Well, this is before knowing about chromosomes and genes. But Mendel reasoned that they had to come in pairs, one from mother, one from father, and we now know that to be true, and we know those are coded for by genes. Principle of dominance came from the P1 to the F1 generation. A trait disappeared, so the dominant trait was the one that masked the appearance of the recessive trait, and his principle of recessiveness came from when we had the F1 to the F2 generation, a trait reappeared. And that meant the trait was present in the F1 generation in terms of a genotype, but wasn't expressed, but was able to be expressed again in the F2 generation. Law of segregation just meant during meiosis, the formation of gametes, that when you make gametes, half of the eggs would have one gene and half the eggs would have maybe the same gene or a different gene, but they would segregate during meiosis, and the same for males making the sperm. Number 4, the law of independent assortment. That applies to dihybrid crosses, trait crosses involving two traits, and all that meant was that one trait's independent of another. You could have one trait and it wouldn't dictate what you had for another trait. Genotype, simply a combination of alleles or genes in an organism. They do the coding for the trait, and the phenotype is what we see. It's how a trait is expressed. Punnett squares, we use these to predict the probabilities for gene pairings. Most of the time you're going to be doing crosses, monohybrid crosses, and you're going to have 4 boxes, and you're going to check out what are the genotypes in those 4 boxes and do it as a probability. Occasionally you'll have to do dihybrid crosses, and in that case you're looking at a cross involving 2 traits, and the result is you end up with 16 boxes filled with genotypes that you have to interpret. We briefly showed you karyotypes, and the karyotypes simply rank the chromosomes in pairs from biggest to smallest. And on the next page we talk about the 22 pairs plus the sex chromosomes. So we'll get to that now. So we know a female has 22 pairs of homologous chromosomes, and they have 1 pair of sex chromosomes, the X and the X. The male, 22 pairs, but now they have the X and the Y was significantly shorter. In terms of sex determination, you can see a female can only chip in an X chromosome, whereas a male, half of the sperm will have the X chromosome and half will have the Y, and that's why the man determines, or whichever sperm gets to the egg determines the sex of the child, and we know that's a 50-50 probability based on Punnett squares for having a boy or a girl. Sex-linked traits are a little more challenging, and that's why we only focus on one kind of sex-linked trait in terms of traits coded for by a recessive gene on the X chromosome. And you have these in your notes, and the two we talked about were hemophilia, the disease where your blood doesn't clot properly, or red-green color blindness. We saw that there are 3 possible genotypes for a female. One can be homozygous dominant, so they have normal blood. Here it's heterozygous, but they still have normal blood. But because they have the recessive gene, they're called a carrier. They can pass that gene forward. And for a female to have hemophilia, to be red-green colorblind, the female would have to have 2 recessive genes. Because a male doesn't have a second X chromosome, all that affects the phenotype for the male is what gene is on that X chromosome. So if they have a dominant gene, they have the normal trait, and if they have a recessive gene, they're going to have hemophilia or red-green color blindness. And once you figure out the genotypes, you can cleverly load them into your Punnett square and then deal with determining probabilities of different types of phenotypes. That's it for part 1.